I cut a piece of pie and put it in the refrigerator for you.
Oh, thank you. How things go on,
Pretty good.
Good.
All right, can somebody confirm you can hear me?
I can hear you.
Thank
I can
you.
hear you.
Thank you. Let me share my screen here. I don't think I'm doing that. Before we begin, are there any questions?
I did have some questions about my grade, but I don't know if you want to answer those now or later.
It'd probably be best to talk about your grade later privately.
Okay.
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 So we can do that after the lecture ends. There is no lab today. Let me go ahead and show you the schedule. Let me get out of that. And today is March 11th. We are supposed to be on chapter 13. We are on chapter 13. We're supposed to finish it today. I don't know if we'll finish it, but we'll try. And let me put a little bit of a break here. The review is going to be Thursday in the lab. Any questions about any of that? All right, so let's get on to chapter 13. There is no lab today, by the way. I will be logging in for the first 15 minutes to answer any questions, but after that, I'll log out. Let's go on to chapter 13 then. I have that we were on this slide talking about virus structure, and we mentioned that viruses have an innermost molecule, their genome, which can be either DNA or RNA. It's never both, it's one or the other. And the DNA can be double-stranded or single-stranded, and so can the RNA. It can be double-stranded or single-stranded. And then lastly, let me remind you, if we have a single-stranded RNA molecule, you need to state whether it's positive single strand or negative single strand. Any questions about any of that? So all viruses have a capsid, that is the protein coat surrounding the innermost molecule. Let me blow this slide up. The capsid is a layer of protein. It is composed of capsomeres. You may have, like this virus only has one capsomere, so one protein. Each of these little circles, let me blow that up even further. Each of these little circles is a capsomere, it's one protein. As you can see, this entire virus is composed of only one protein. And of course, the genomic molecule. Some viruses are that way. Other viruses, like this one here, will be composed of at least a couple of different proteins in the capsomere. Let me shrink that down even further. There we go. And I'll show you one here. Sure, that's it. No, it's this one here. This virus here has several proteins. It'll have at least one protein in the head, at least two in the sheath, meaning the inner core and the outer core, and then one protein, at least for the tail fiber, probably a different protein for the tail pins, and then a different protein for the base plate. Sorry, at least probably two proteins for the base plate, one for the pin and then one for the base plate. Any questions about any of that? Where were we? So if a virus only has the genomic molecule and the capsid, meaning there's no envelope, we call it a naked virus. Some viruses have an envelope. This is a lipid bilayer. It does have some proteins, carbohydrates in it, but it's mostly phospholipid. This will be external to the capsid, and so it'll be surrounding the capsid here. And there we have one there. You can kind of see it if I blow that up. There's the genome, there's the capsid, and there is the envelope. The envelope may be covered with glycoprotein protein spikes, shown here, the reddish purple spikes. Come on, mouse. That are attached to the envelope. The spikes, if they're there, they are antigens, meaning they project from the surface of the virus, and they are the externalmost molecules which the host immune system is most likely going to recognize and then bind to. So these molecules serve as antigens to the host that the virus infects, assuming it's an animal. And the spikes are also the most external molecules. So they also are the molecules which bind to the receptors, meaning the spikes have the adhesions or the ligands of the virus. So they're both the antigens to the host and then the ligands or adhesions to the virus because of the outermost molecules of the virus. Now, on this virus here, which is a naked virus, where are the adhesion molecules? Where are the molecules that bind to the host cell receptors? Anyone have a guess? Well, on this one, it's pretty easy. There's only two molecules in the virus, and that's the genome and the capsomeres, the molecule that makes up the capsid. Anyone have a guess now? All right, well, think about that. When we're talking about viral structure, it's important to talk about the virus morphology. This is the shape of viruses. We can classify a virus based on their capsid structure. We have helical viruses shown here where the capsid is in the structure of a helix. We have polyhedral structures or shapes of virus where the virus is actually like a many-sided complex dice, a polyhedral. And there we have an actual electron microscopic image of a polyhedral virus. And then the third category is a complex virus. And this virus can take on almost any shape. Here we have a complex virus with an envelope. There's another complex virus. I don't even know what shape that would be. Whatever shapes those are. Something like a ellipsoid for this one. And then there we got a complex virus, the T4 bacteriophage, which is the virus which is most studied of all viruses. And it looks like the Apollo lunar lander, that type of a shape. Any questions about any of that? Let's talk a little bit more about bacteriophage T4 because it is the virus that has been studied the most. The reason why I think it's been studied more than any other virus was bacteriophage T4 infects E. coli cells, and every microbiology lab happened to have E. coli in it, so they naturally studied T4 because it was fairly easy to grow since they had E. coli, and it just infects E. coli. T4 belongs to the T-even bacteriophage family of viruses. They all have this shape. And then there are T2, T4, T6, their names. T4 is the one we're going to talk about, taking on this shape here. It does have its genome. It's in the head. This is this portion of the virus there. And then there are tails to some viruses, particularly bacteriophages. That's because bacteria have a cell wall, and it's more difficult to infect a cell when it has a cell wall than when it's lacking a cell wall. And the tail is a specific structure to help with infecting a bacteria that has a cell wall. Now, not all bacteriophages have a tail, but many of them do. The adhesions or ligands, the molecules of the virus that bind to the host cell receptor, are on the bottom of the tail fibers. So this is where the virus binds to its host cell. Infection with a virus in an animal cell tends to stimulate the host to produce antibodies that then react with, bind to, and attack the virus. Antibodies recognize and bind to antigens, and that would be the viral surface proteins. Some viral proteins can change or mutate frequently. Therefore, different viral strains can therefore infect the same individual more than once. An example of this is COVID-19. There are reports of some people being infected at least 6 times with COVID-19. And the reason is because COVID-19 tends to change and mutate fairly frequently. And so new strains come out, and then they can infect the same individual as what an earlier strain infected. Another good example is influenza. This virus mutates fairly rapidly and then can change about once every 10 years. And every 1 in 10 years it's a new virus. And so the people's immune system have never seen it before. And then we have an epidemic because there's no immunity to the new flu virus. But it also changes fairly rapidly by mutation. And so when you get a flu shot, they say that after 3 years, you're no longer protected against the flu from the flu shot. Now, in reality, that's an oversimplification. The immunity would be, this would be a percent immunity up here. It never reaches 100%, but in young adults and teenagers, the immunity is quite high. It's over 90%, so it's around 95%. And immunity over time with the flu just decreases. And after 3 years, the immunity drops below a threshold where we say you're no longer protected anymore. And obviously you have some protection, it's just reduced protection. But over time, you will eventually get immunity where the flu shot will be 0%. I don't know what that is, but 3 years is below a threshold where they say the flu shot is on average not protecting you anymore. And that's for an average individual on an average year and an average flu, all that sort of stuff. So that's why you need to have the flu shot at least once every 3 years. And because the immunity decreases, they do recommend you get the flu shot every year. Any question about any of that? And COVID-19 is a little less mutating than the flu, but it's close to that. So you should get the COVID-19 shot at least every 2 years, if not every year. I suppose it depends on your age too, but what am I trying to say here? Oh, now not all viruses mutate this rapidly, where the flu gives no protection, essentially no protection, after 3 years of getting the flu shot, just from mutation of the virus. But then again, there's other viruses which we made vaccines in the 1950s, like polio, for example. Those vaccines are still used today. So polio has not changed since at least the 1950s. And what is that? That's about 70 years ago. So, like I said, some viruses mutate frequently and the immunity is not, well, the immunity to the new strains is not any good. And then others don't seem to mutate that rapidly. And like I said, the vaccine are still good, made in the 1950s or earlier. I think yellow fever was made before 1950. It was made in like 1940 or something like that. And that vaccine is still used today. Any question about any of that? All right, let's talk about viruses, viral taxonomy. Viruses are grouped into families based upon their type of nucleic acid, the replication strategy, their morphology. We do have viruses in a category above family. Let me see if I can remember what that was. King Peter's son came over from, so it's orders. So we do have viruses grouped into orders, but above an order, we do not have viruses into groupings. The exception is we do have viruses grouped into the family of not the family, the kingdom that they infect. Like there's animal viruses and there's plant viruses and there's bacterial cell viruses. So we do group them into the kingdom they infect. But everything else, like the subphyla, the phyla, whatever, we don't group viruses into them because we do not know which phyla they belong to. And the reason is viruses have a very small genome. They're not terribly large, and their genome evolves very quickly with the host cell that they infect. So viruses do evolve quickly with their host cells, and with their small genome, we just don't know the evolutionary history of viruses. And so we don't group them anything much above an order. Any questions about any of that? But we do group them into families, and then into genuses, and then into viral species, and then subspecies underneath that. A virus family name will end in viridae. So when we're talking about the herpes family of virus, I will use the name herpes viridae. The genus name ends in virus. So for the herpes genus, it would be herpes virus. For the viral species, this will be a group of viruses sharing the same genetic information, an ecological niche. We use common English names. For a herpes virus, let me think about it. We'll talk about herpes simplex virus. So that would be the species. And then there are subspecies. They're designated by a number, and there's herpes virus, herpes simplex virus type 1 and herpes simplex virus type 2. So those are two different subspecies of the same species of virus. Any question about any of that? All right, with the viral names, there are no Latin or Latinized names. So anywhere you go in the world, if you use the term poliovirus, everyone will know what virus you're talking about. And if you use the word herpes simplex virus, everyone will know what virus you're talking about. And it's surprising that all of the common English names are English. It turns out that the original, the first virologists were all English speaking, and they simply gave the viruses common English names. And then when virologists appeared in the other languages like French and German, they just adopted the English names that the viruses were already named. And so these common English names are actually used worldwide. Any question about any of that? Let's talk about how important the subspecies is. Actually, you already know that herpes simplex type 1 and herpes simplex type 2 are really different in the diseases they cause. Herpes simplex type 1 tends to cause cold sores around the, usually the mouth or the nose. And then herpes simplex type 2 causes not cold sores, sores around the genitals. That isn't entirely true. You can get cold sores on the genitals, and you can get herpes simplex type 2 sores around the mouth. So this is a generality, but that's the way most of the cases are. But herpes simplex type 2 can infect the nose and the mouth, and herpes simplex type 1 can infect the genitals. But they tend to go the other way. A more important distinction between the subspecies is, let's talk about the poliovirus. We have poliovirus subspecies 1, and it causes the disease that we call polio. On the other hand, the poliovirus subspecies type 2 is the vaccine that we use to vaccinate people against polio. So one subspecies is used as the vaccine to prevent polio, and the other subspecies causes the disease polio. Any questions about how important the subspecies is to viruses? All right. I've already gone through that, so let's move on. Growing bacteriophages in the lab. Actually, I've done this where I grew bacteriophages in the lab. It's fairly easy to do. First, you get a plate and you coat it with a bacteria strain that'll grow the virus, something like E. coli if you're trying to grow bacteriophage T4. And then you streak it everywhere so that you get a lawn of bacteria, meaning the bacteria will grow everywhere. And that's why this plate is mostly whitish from the bacteria growing as a lawn of bacteria cells everywhere. And then you get a solution containing your bacteriophage and you smear it over the plate everywhere. And wherever there is a virus that grows on the bacteria, you then incubate the plate, getting the bacteria to grow as well as the virus. And then you pull out the plate and you see plaques on the plate. Each plaque is where a bacteriophage infected a bacteria cell, reproduced in the bacteria cell, and then went on to infect neighboring bacteria cells, forming what we see here, a plaque. And a plaque is just where there are no bacteria because the virus has lysed the bacteria. Any questions about any of that? Now this is probably 3 plaques that grew together, but that one is a large plaque. That one's fairly large too. Well, let's compare this one with that one there, a very small plaque. Probably what happened was this bacteriophage infected a bacteria immediately, reproduced, and then lysed the cell, went out and infected more cells. And so this is a large plaque because it got started right away. On the other hand, let me get this, well, let me use my phone here. On the other hand, this virus here, let's say it went right here. I don't know if you can see this. And then there's a bacteria cell nearby, but not where the virus was. And what happened was the virus then grew and then grew and then grew and then grew one more time. And now the bacteria cell is touching the virus. And so that's probably why this plaque right there is smaller than that plaque right there. And that is the plaque did not start to grow immediately. It needed the bacteria cells to grow until the bacteria cell finally touched the virus, the bacteriophage, and then started the formation of the plaque. So that's probably why that plaque is smaller than that plaque. Any question about any of that? All right. So when we're talking about viral multiplication, you should come to understand that the viral genome only contains a few genes. And it does contain the genes for the capsid proteins needed to make the coat of the virus. And virus replicate by hijacking the cell and then forcing the cell to replicate the virus. Following infection, viruses take over the host cell's metabolic machinery and then use that machinery to replicate the virus. The viruses used host cells' ribosomes, host cells' tRNA, host cells' amino acid, host cells' ATP, and some of the host cells' enzymes to replicate the virus. The basic mechanism for viral multiplication is similar in all viruses. For bacteriophages, we have only 2 life cycles that a virus can replicate in, meaning 2 mechanisms the virus can replicate in. Viruses, once they infect a bacteria cell, they can only replicate in one of 2 ways. First, there's the lytic life cycle, where the host cell is infected. It'll be used to replicate the virus. It'll be lysed, and that'll kill the host cell. And then when the cell is lysed, that will release the newly replicated viruses, the virions. And then there's the lysogenic life cycle. When the virus infects the host cell, the virus and the host cell will live in symbiosis for as long as the lysogenic life cycle remains. So in the lysogenic life cycle, the host cell is infected with the virus, but the 2 are living together. Any questions about any of that? All right, in making a new bacteriophage, what do we need to make new virions? What components need to be made? Well, if we have a double-stranded DNA virus, we have to replicate the genome. And that's true for whatever the genome is. You need to replicate the genome. And that'll get done by DNA replication. And then you have to transcribe the genome to get viral messenger RNA, and then translate the viral messenger RNA to get viral proteins, particularly the viral capsid proteins. And then the viral capsid proteins need to come together with the replicated genome to make the newly made progeny virions. Any question about any of that? All right, in the lytic life cycle of a virus, the virus goes through 5 stages of its life cycle. First, there's attachment, then there's penetration, then there's biosynthesis of the virus, there's maturation of the virus, and finally release of the virus. Let's watch a little video about the viral.
First, the phage particles stick.
I think what I want to do is show you another movie because this movie doesn't show you anything. And it's because the player here just doesn't read the movie correctly and it's only discussing it but not showing it. Maybe you might have another player which would work. I need to get, what's that player, VLC or VMC on this computer, and I just haven't done that. I think this is the one I want to do.
In a way, a virus is a parasite. It attacks a host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell. This process of viral multiplication is called the lytic cycle.
All right, let's stop there and we'll continue this with the lysogenic life cycle in a little bit. Any questions about the movie?
I have a question. My son actually just asked, are there any viruses that are good viruses?
Well, poliovirus type 2 we use as a vaccine to prevent getting polio. So I guess you could call that a good virus. If you mean, are there any viruses that benefit humans?
Yes.
Well, they think, and this is stretching it a little bit, but they think that humans partially evolved because the virus got into our genome, and the virus is in our genome, but it's non-replicating, meaning a non-functional virus. And I think humans have 5 viruses in their genome, all of which are non-replicating, non-functional viruses, but they make up part of our human DNA. And it's thought that humans evolved partially because of that. So there's one good aspect, I guess, of that a virus has done. There were at least 5 times in the past a virus that infected our genome and then became a part of our DNA. Any question about any of that? All right, and like I said, that's a little bit of a stretch because that's assuming that humans evolved because of it. And I think there's some evidence, but obviously it's not strong evidence because you're looking at something that happened probably millions of years ago. And when a virus normally infects a person, it obviously doesn't benefit the person. Okay, so normally when a virus infects a person, it normally does not benefit the person, with the exception that, like I said, we do use some viral strains as vaccines, and there are more than one example of that. Any live vaccine is a living virus that we're using as a vaccine. And I think it's the Sabin vaccine for polio, and then the yellow fever virus. That is a living virus that we infect into people to prevent them from getting yellow fever. All right, any questions about that? All right, let me, I guess I don't want to end that because I want to continue the movie later. Go back here. So first there's attachment where the virus bacteriophage actually attaches to the host cell. And like I said, there are adhesions or ligands on the virus, specific molecules that bind to specific molecules on the host cell that we call host cell receptors. And that's how attachment of a virus happens to its host cell. And then there's penetration. That's where the virus opens up the cell wall. And that, let me go ahead and show you a little bit of that right here. What happens is the virus actually secretes the enzyme lysozyme, which right here will then digest the cell wall right there. Any questions about any of that? In the movie, it had the base plate rotating. I don't know if that happens or not, but the rotating base plate does not eat a hole in the cell wall. It's actually the enzyme lysozyme, which eats a hole in the cell wall. And then the virus contracts, which is shown here. This is a little extended there. And what happens is the tail fiber contracts, or the sheath contracts, and that puts pressure on the head, and that will inject what's inside the head into the cell through the cell membrane, and that will inject the viral genome into the cell. And that's penetration. Then there's biosynthesis, where the virus takes control of the host cell, uses the host cell to replicate the virus, both the genome of the virus and the viral proteins. And that's actually a little complex. Let me say, yeah, it's not shown here. I don't remember if it was showed it in the movie or not, because I went to get a drink of water, so I didn't watch the movie. But what happens is usually the virus will secrete an enzyme, which then dissolves the or cuts up the host chromosome. And the reason for doing that is the viral genome wants to call the shots in the host cell it infects. And an easy way to call all the shots is you digest the host chromosome, and then your genome is calling all the shots in the cell. And then there's viral assembly, where the viral genome will be replicated. For a double-stranded DNA virus, is pretty easy. You just use each strand as a template to make a newly replicated strand. And then you translate the or transcribe the viral messenger RNA, and then translate that into viral proteins. And then the viral proteins actually self-assemble into a virion. And then they have an enzyme which actually stuffs the viral genome in the head of the virus, I think before it's assembled. And then the new virion self-assembles. The last thing the host cell, the virus does, not the host cell, the virus genome does is it secretes an enzyme, lysozyme, which then dissolves the cell wall of the bacteria, and then water can enter the cell, and the cell membrane will expand because there's no cell wall, and eventually the cell will burst, and that will release the newly replicated virions, which is actually here. Any questions about any of that? All right, there are two terms you need to know in the lytic life cycle of a virus. A virus that undergoes the lytic life cycle has a burst time and a burst size. The burst time is the time from initial infection of the host cell till the time where the host cell bursts. For an average virus at an average temperature, it'll take 20 to 40 minutes, depending on the species of virus, as well as the temperature you're growing the virus and the host cell at. So it is temperature dependent as well as strain of the virus dependent. And then the burst size, the number of newly replicated virions that are released, are usually between 50 and 200. This will depend mostly on the the strain of the virus. Any question about any of that? All right, so that's the lytic life cycle. Let's talk about a lysogenic virus. So this is another bacteriophage that when it infects the bacteria cell, it can enter the lysogenic life cycle, in which case both the virus and the bacteria host cell will live in symbiosis. However, once a lysogenic virus infects a host cell, it immediately has to decide which life cycle to enter, meaning a lysogenic life cycle virus can enter the lytic life cycle, or it can enter the lysogenic life cycle. And it has to decide this immediately once infecting a host cell. If it enters the lytic life cycle, then we just follow the lytic life cycle that I just talked to you about, meaning the virus injects its genome into the cell, immediately decides to enter the lytic life cycle. It replicates the virus, lyses the cell, releases the new virions. However, the virus can choose to enter the lysogenic life cycle, and it will, in the lysogenic life cycle, not lyse its host cell. The virion, the virus, and the host cell will live together in symbiosis for as long as the lysogenic life cycle continues. What happens is the bacteriophage lytic life cycle genes are suppressed, so they go latent, meaning there are genes in the lysogenic life cycle that are important in replicating the virus in the lytic life cycle. These genes are suppressed while the virus stays in the lysogenic life cycle. What happens is there's a regulatory protein that shuts down the lytic life cycle, and while that's shut down, the lysogenic life cycle continues. So it remains in the lysogenic life cycle as long as that repressor protein is active. Any question about any of that? All right, so let's talk about the lysogenic life cycle. Like I stated, once the lysogenic virus infects the host cell, it immediately has to decide whether to enter the lytic life cycle or enter the lysogenic life cycle. So let's watch a little movie on that, where the virus decides to enter the lysogenic life cycle. I think this is it here.
Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates, and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
All right, any questions about that part of the movie? So to enter the lysogenic life cycle, the genome of the virus integrates into the host cell's chromosome. This we call a prophage, where the viral genome is a part of the host cell's DNA. Now, whenever the host cell replicates its DNA, it replicates the viral DNA. And that way, when the cell replicates into two, each cell, daughter cell, will get a copy of the prophage. And this can continue for many generations where the lysogenic life cycle continues. Like I said, in the lysogenic life cycle, the virus can replicate, but only when the host cell replicates its own DNA. Any questions about that? All right. And then for reasons which are not totally clear, every once in a while, the virus can leave the lysogenic life cycle and enter the lytic life cycle. If it does that, the prophage is excised from the host cell's chromosome. And then the virus enters the lytic life cycle where it replicates the viral genome and then replicates the viral capsid protein. The viruses self-assemble, and then the cell will be lysed, and that will release the newly replicated virions. Any questions about any of that? All right, so it's not entirely understood why the lysogenic life cycle ends and the lytic life cycle begins. And they've studied this now for over 70 years, and they still don't have a good understanding why the lysogenic life cycle ends and the lytic life cycle begins. They do know one factor which can cause it, and that is if you damage the host cell's chromosome. For some reason, the virus seems to know that the host cell's chromosome is damaged and that the cell may die, in which case the virus leaves the lysogenic life cycle and enters the lytic life cycle. But other than that, we don't know why the virus leaves the lysogenic life cycle and enters the lytic life cycle. Even after 70 years of studying the this aspect. Any question about any of that? All right. Let me see if there's anything I need to talk about. I've already talked about prophage, the term used when the viral genome is a part of the host cell's chromosome, and that there's a repressor protein that is active in the lysogenic life cycle. It keeps the lytic life cycle genes dormant, not expressed. And that can go on for many generations. And then the lysogenic life cycle ends when the prophage is excised from the chromosome, and then the lytic life cycle will begin. All right, there are some consequences to lysogeny. First, a lysogenic cell, meaning a host cell that has a lysogenic virus in it, is immune to infections from a virus of this species of bacteriophage. So once you have one copy of the lysogenic virus in you, the host cell is immune to further infection by that species of virus. And then the second consequence of lysogeny is this host cell can pick up new DNA, which it can express new DNA, altering the phenotype of this cell. The new DNA is the viral genome, bringing in new DNA to the host cell. And that DNA can be expressed, changing the phenotype of this host cell. Any question about any of that? Some examples of a virus bringing a gene to a host cell, changing the phenotype of the host cell, and this is called phage conversion, is Corynebacterium diphtherium. This bacteria, Corynebacterium diphtherium, produces the diphtherium toxin only when the bacteria is infected with a lysogenic bacteriophage. So when the Corynebacterium diphtherium does not have the virus in it, the lysogenic virus in it, it does not produce the Corynebacterium diphtherium toxin. Only in an infected cell, infected with a lysogenic virus, can the Corynebacterium diphtherium produce the diphtherium toxin. Any question about any of that? All right, enough talking about bacteriophages. Let's talk about animal viruses. Animal viruses are viruses that infect cells in the kingdom animal, and they follow, surprisingly, the basic pattern for phage multiplication as bacteriophages, but there are some differences. First, the attachment is a little different because animal cells don't have a cell wall, they only have a cell membrane. And that actually makes it easier for the virus to infect the cells. Penetration is a little different. It happens by either endocytosis or by fusion. We'll talk about that in a little bit. Encoding is about the same, but synthesis is very similar to bacteriophages. Maturation also similar. And then release can differ. We do have animal viruses that do follow the lytic life cycle, so they will infect a host cell, replicate the virus, and then lyse the host cell to release the newly replicated viruses. So we do have the lytic life cycle in animal viruses. We do not have the lysogenic life cycle viruses, but we do have the budding life cycle virus. And these viruses bud out of the cell. And these viruses are envelope viruses. And they usually replicate at low numbers, so they do not kill their host cell. And the host cell remains alive and will just continue budding the viruses for as long as the viruses are replicating. But the viruses are replicated at low number. And we call that budding. Any question about any of that? All right. So the multiplication of animal viruses follows the same general pattern as the multiplication for bacteriophages. But there are some differences. The most important difference, the most major difference, is bacteriophages can only replicate in the cytoplasm because that's all bacteria have. However, animal viruses can replicate in the cytoplasm or they can replicate in the nucleus. So that's a major difference. The attachment is also different in that animal viruses attach to the cell membrane. Animal cells don't have a cell wall. The attachment and then entry into the cell happens by either penetration, by endocytosis, or by fusion. So the earlier copies of the textbook actually had this wrong, where they said that the entry of the virus into the cell, they initially said it occurred by penocytosis. That's in the earlier copies of your textbook. If anyone has an earlier copy of the textbook, just make a note of that. If you do not, then your copy of the textbook is correct, and that is that entry of the virus can happen by endocytosis or by fusion. Endocytosis is where the virus attaches to the receptor of the host cell, and then the host cell begins vaginating, the cell membrane begins vaginating around the virus, and will eventually happen is this will be brought into the cell by the cell membrane, pinching off right there, reforming the cell membrane, and then forming a vesicle membrane right here around the virus. And that will bring the virus into the cell by endocytosis. Any question about any of that? All right, the other way that viruses can be brought into the cell are by fusion, and that is we have a cell membrane, which is a double layer, which is why I'm doing two here. Cell membrane is a phospholipid bilayer. And then there's an envelope virus. We're going to make it here. And it's enveloped with a lipid bilayer right here. And I'm just going to make this virus the shape here. So this would be the capsid. And then there's the genome of the virus. The bilayer of the cell membrane and the bilayer of the envelope of the virus can fuse. I actually got those together and those together. That's pretty good. I managed to do that. And let's see if I can get this to work. Oh, let's make that a little wider over here. Make it a little wider here too. When they fuse, trying to make that a fuse. Trying. There we go. It's a little narrow, but you're getting the picture. Where the virus is now open to the cytoplasm, and this can move through here. You got to make this a little bigger, but let's go down here. The virus can now move into the cytoplasm. Any question about any of that? That's membrane fusion, cell membrane fusion of the envelope fusion of the virus. And why they can do that is they're both phospholipid bilayers, mostly made up of phospholipid. All right. Let me talk a little bit about this one again. Let me move back up a little. Right here. Some viruses can replicate in the nucleus, other viruses in the cytoplasm. As a rule of thumb, the DNA animal virus replicates in the nucleus. Why? Because in the nucleus of an animal cell, we have all of the ingredients for replicating DNA. And so the DNA virus replicates in the nucleus. And that's a rule of thumb. And I don't know of any exceptions to that rule of thumb. For an RNA virus, they, on the other hand, replicate in the cytoplasm. And the reason why is it's in the cytoplasm, we have all of the machinery for translating and making protein, which is needed for replicating RNA virus. And so RNA viruses, as a rule of thumb, replicates in the cytoplasm. There is one exception to that rule of thumb, and that is there are retroviruses, which are RNA viruses, and they replicate in the nucleus of the cell. They do not replicate in the cytoplasm. And they're the only exception I know of to the rule of thumb. Any question about any of that? All right, let's talk about the multiplication of animal viruses. As I mentioned, we do have animal viruses that do follow the lytic life cycle. Poliovirus is one virus, animal virus, that follows the lytic life cycle. I'm not sure, but I think the human cold also follows the lytic life cycle. Like I said, don't quote me on that because I'm not an expert on the human cold virus, but I think it does follow the lytic life cycle. And polio, I know, does follow the lytic life cycle. There are other animal viruses that follow the budding life cycle, and they replicate at low numbers when they replicate, and replicate following the budding life cycle, where the new virus gets together with its capsid protein. So this is the naked virus. And a budding virus is always an envelope virus. And the new virion comes up to a membrane and begins pushing on it. That can be either the cell membrane, the nuclear membrane, or the membrane of the endoplasmic reticulum. And that virus then forms a bud, forming a bud in the membrane that we're talking about. And then that virus can rip off the bud where the envelope reforms. So if that's the cell membrane will reform, and then a envelope forms around the virus. And that's why a budding virus always has an envelope, because it buds out of a membrane, which is a lipid bilayer. And that's why the envelope is mostly phospholipid, because it is a envelope, either the envelope of the cell membrane, the envelope of the nuclear membrane, or the envelope of the endoplasmic reticulum, that is where the phospholipids came from. Any question about any of that? Now, the envelope tends to have viral proteins and glycoproteins in it, and what happens is the virus codes for the proteins and the glycoproteins in the membrane we're talking about, and then puts them there. And for some reason, that virus knows to come to a part of the membrane that has the viral proteins and glycoproteins in it. And then it tends to rip off a portion that has the viral proteins and glycoproteins in it. Any question about any of that? I moved that by accident. All right, so that's the budding life cycle viruses. The budding life cycle viruses always have an envelope. And then the lytic life cycle viruses are always naked viruses. There's two more life cycles I need to talk about. I'll talk about the latent life cycle later, but I do want to briefly mention there is a life cycle very similar to the budding life cycle. It's not the budding life cycle because the virus doesn't bud out of the cell or the membrane of the cell, and it is a naked virus, and we call it a persistent infection life cycle, where the virus can be shed from the cell for a long period of time, what's called persistent infection. Usually the virus is shed at low numbers. What happens when the virus is replicating is it's a naked virus, and it gets put inside of a vesicle. The vesicle then fuses with the cell membrane of the cell, and then this, when it does, it opens up, releasing the contents of the virus. Oops, go back to the outside. So what happens is, let me do this in paint quickly. We have a cell here, and we make a virus here, and let's say there's several of them, because usually there'll be more than one. And it gets put inside a vesicle. The vesicle then comes up to the cell membrane and fuses. When it fuses, let me draw this over here. When it fuses, the cell membrane now goes around the vesicle, so it's part of the vesicle membrane, and then comes back here, and that opens up the vesicle to the outside, dumping the viruses outside the cell. So it's similar to budding, but it's not budding because that isn't budding out of it. It's the vesicle that is fusing with the cell membrane, dumping the contents, which are a naked virus, out of the cell. And we're not going to really talk much about the persistent life cycle, persistent infection life cycle. I'm just stating that there is that life cycle that's similar to budding, but a little different. Mainly, it's a naked virus, not an envelope virus. All right, so viruses have different genomes. Depending on the genome, you have to have a different replication strategy. So there are different mechanisms for multiplying the viral genome. Let's talk about the DNA family of viruses. There are 7 DNA family viruses that infect animal cells that cause disease in humans. So there are other animal family of viruses, but they don't cause diseases in humans. There are only 7 that cause diseases in humans, and I'm showing you this here. You don't need to know the names of them or the type of the strand of DNA it is, whether it's an envelope or naked, the capsid symmetry, the size of the virus, nor do you really need to know the diseases. I'm just showing you that. We will talk a little bit about pox viridae, because that's the family of DNA viruses that smallpox is a member of. And we've already talked about herpes family of viruses. So we've already talked about that. And that includes shingles and chickenpox, that family, cold sores, genital sores for herpes simplex type 2, cytomegalovirus, which I haven't talked about yet, mononucleosis. So some common human viruses in the herpes family. The papilloviridae, I'm not going to talk about that family. I'm not going to talk about adenoviridae. Let me talk about a little bit of that one now. This family is a family of viruses that actually cause the human colds. There are 3 families of viruses that cause the human cold. This is the least numerous and least common family that causes the human colds. So there's 2 families of RNA viruses that cause human colds that are more common than the adenoviridae. But this is one of the 3 families that cause the human cold. And I'm not going to talk about the other families either. Any question about any of them? All right, so we've talked about the herpes family of viruses. For DNA-containing viruses, they generally replicate in the nucleus. capsids and the proteins are synthesized in the cytoplasm. And so the capsid protein coat has to get together with the replicated virion to make the newly replicated virions. Any question about any of that? So once viral DNA enters the host cell, one of two things can happen. First, the DNA can actually integrate into the host cell's chromosome. We don't call this a prophage because a phage is a term we use for bacteriophages. We call it a provirus. This is an animal virus that is integrated into the host chromosome. And once this integration happens, it'll be permanently in the host chromosomes, meaning it is never excised. It will continue to be in the host cell for as long as the host cell is alive. And if the host cell divides into two, it will replicate the provirus, and then both copies of the both daughter cells will get the provirus. So once this happens in an animal cell where the virus genome integrates into the host cell, it becomes a permanent fixture of the DNA of that host cell. The other thing that can happen when the DNA virus infects a host cell is the viral DNA can be expressed, making viral messenger RNA, which will then move into the cytoplasm to make viral proteins. The viral DNA can be replicated to make new genomes, and then the viral genome will get together with the viral capsid proteins to make the new progeny virions. Any question about any of that? How are we doing time-wise? I always have time to do this. All right, let's talk about multiplication of DNA viruses in more details. We have a virus which infects a host cell, and what happens is there will be adhesion molecules or ligands on the virus which will bind to the host cell molecules that we call receptors. And then the virus will get into the cell, either by endocytosis or by fusion of the envelope to the cell membrane if the virus has an envelope. The virion then enters the cell, will move into the nucleus. It will make a messenger RNA copy of the viral DNA. The viral messenger RNA will move into the cytoplasm to translate viral proteins, especially the capsid proteins of the protein code of the virus. The viral genome itself in the nucleus will replicate, making copies of the viral genome. The viral genome will then get together with the capsid proteins to make the newly replicated virions. And then the virus will be released from the host cell, either by lysis of the host cell if the virus follows the lytic life cycle, or by budding if the virus follows the budding life cycle, or by what do you call that? Exocytosis, if it is similar to the budding life cycle, but is a naked virus that we're going to call a persistent viral infection. And you don't need to know about the third one. We won't talk about it. But the point is the virus will be released from the host cell, and then it can then infect another host cell, causing the process to begin all over again. Any question about any of that? All right, if not, we need to talk about the RNA family of viruses. And we're looking at them here. There's 15 families of RNA viruses that infect human cells and cause disease. So you don't need to know about the disease here, but those are the family names of the 15 families here. and then the type of genome it is, and whether the virus is naked or not, capsid symmetry, and then the size of the virus. Like I say, you don't need to memorize this table. We will talk about a few of the virus families, like family Picornaviridae is a very large family of RNA viruses that infect human cells and cause various diseases. In the family Picornaviridae, it is the #1 cause of human colds in humans. So the most human colds are caused by the RNA family Picornaviridae. Any questions about that? We'll talk about the #2 in a little bit. But this virus also causes, or this family of virus also causes other diseases. This is the family that poliovirus is a member of. It's also a family that hepatitis A is a member of. And so you can see this family is very diverse. And like I mentioned, it's already a very big family. Very different between a rhinovirus, which causes human colds, hepatovirus, which causes hepatitis, and polio, which causes the disease polio. We're not going to talk about that one or that one. But we've already talked about, oh, never mind, that's not herpes. That's herpes. Astroviridae, Togaviridae, yeah, we won't talk about those. Coronaviridae, we will talk about that one. I've got 3 slides on this one because it's the family of viruses that causes COVID-19. This family was actually well known to humans long before COVID-19 ever evolved in 2019, or infected human cells in 2019, because this is the second most common cause of human colds. The family Coronaviridae is well known to being the second most cause of human colds. Retroviridae, we'll talk about that. Retroviruses like HIV. I won't talk about Paramyxoviridae, which is too bad because that's a lot of human, especially childhood illnesses. Orthomyxoviridae, I do want to talk about that just a little bit. This is the family of viruses that cause the flu. And I guess that's all you need to know. Let me shrink that down so I can go down here. And we're not going to talk about any of the others. Okay. Any questions about any of them? All right, here are some RNA containing animal viruses. You can note they have different shapes, different sizes. They cause different diseases as well. RNA family of viruses in general replicates similar to DNA viruses, with obviously some differences because they have RNA for their genome, not DNA. RNA family of viruses, as a rule of thumb, replicate in the cytoplasm. There is one exception to that rule of thumb, and that's the retroviruses. They replicate in the nucleus. That's the only exception that I know of. When we're talking about single-strand RNA genomes, you need to differentiate between a positive single-strand RNA and a negative single-strand RNA. The positive single-strand RNA genome can be read by the ribosome and be translated into protein. The negative strand RNA genome cannot be read by the ribosome, and it cannot be translated into protein. Any question about any of that? All right, I think we'll stop here and I will continue with this next time. I will be in the lab from 6:30 to 6:45, but only for 15 minutes if you have any questions. All right, I'm not going to log out, so hopefully everyone else will log out. There was a student who wanted to talk to me about their grade. Hopefully everyone else will log out. If not, we'll have to log into the lab. But let's see how many people I have.
I don't mind if anyone